Next company up is PurposeBio. Lital?
Hello, everyone. I'm Lital. Nice to meet you, PurposeBio. We are a UCLA startup out of Elaine Shao's lab. She's my co-founder and the scientist. And according to UCLA, we are the best, one of their best and most investment-worthy startups, worth mentioning. What problem do we solve? Cardiometabolic disease. We are in synthetic biology platform developing a first-in-class therapy for massive unmet needs in cardiometabolic disease, such as NAFLD-NASH and hyperlipidemia in patients that are intolerant for statins. Our first indications, as I said, are hyperlipidemia and NAFLD-NASH. We have a rich platform pipeline opportunity in the future, but we have chosen to focus on the two largest hanging fruits and unmet needs currently. So in hyperlipidemia, I'm sure you're familiar with the miracle drugs, statins, great drugs, saving lives every day, but in fact, about 50% of the patients that need statins cannot use statins because they're either intolerant for them or discontinue them due to adverse side effects. We target this entire pie, both the patients that are intolerant to statins and patients that do take statins, as we believe we can manufacture and develop the next generation of statins. In terms of fatty liver disease, a huge unmet need, probably the Western world's most growing epidemic, hitting about 30% of adults with no FDA approval. And if you're following the news, there's been a big failure, unfortunately, in the market just last week. So what do? We have identified a very interesting enzyme in gut bacteria that has a very important role in lipid metabolism. It targets secondary type bile acids, and secondary type bile acids are the upstream mechanism in lipid metabolism between the liver and the gut and the small intestines in a circulation called enterohepatic circulation. So up until today, it's known that secondary type bile acids are the ones that will cause disease, that will cause fat to accumulate in the liver and triglycerides go up in the blood and cause cardiovascular and fatty liver disease. Unfortunately, up until today, no one was able to target only secondary type bile acids, and our platform is the first and only one to target these bile acids. We have in vivo data, preliminary data. We are showing that we're reducing triglycerides, cholesterol. We're even reducing fat, but we're not a fat weight company. We're a therapeutic company. And most importantly, we're precisely targeting only secondary type bile acids to, if not prevent, but definitely treat disease. We also show in mice that we're significantly shrinking adipose tissue size. These are the fat cells in our blood. A very important fact to explain is that bile acids are upstream of mechanisms such as FXR. These are sensors in the liver. The gold mine of pharma in the last few years is to try to find the NAFLD-NASH therapeutic, and many of the companies are targeting FXR sensors. Bile acids are controlling these sensors. These are upstream of FXR, and we believe are the main right proper target for fatty liver disease. This is our amazing team. I started PurposeBio out of an EAR program I did at SRI. Professor Elinav from the Weizmann Institute, he's considered to be one of the Bill Gates in our field, and he's the chair of our scientific board. Dr. Elaine Shao already spun out technologies to clinical phase companies, and the rest of the team. We have raised, we have a commitment of $1 million. We've raised 250. We are engineering our enzymes as we speak. And our next milestone is to test our engineered enzyme that will have a therapeutic effect with higher activity and stability in disease mouse models. This would be in about 9 months from now. I have a lot more to say, but it's only 3 minutes.
Great. Judges?
I'll go. Thanks for the presentation. I have a question. The enzymes are found in gut bacteria?
Yes,
Naturally.
naturally. So
You're boosting the gut bacteria. You're boosting the enzymes in some ways.
boosting the enzyme. So the progress of the science was Elaine has studied this understudied species, gut bacteria, and she found that it has some effects on lipid metabolism. So the data out there is very much supporting what we see in the lab.
And basically you're doing synthetic biology.
Yes.
And how is the manufacturing? Is it aerobic, anaerobic?
It's an anaerobic strain. We have identified our preferred manufacturer. It's a company called Bios in France. There are actually two large companies that I would trust with manufacturing an anaerobic strain, and that's Bactera out of Lonza in Switzerland and Bios in France. These are the two leading ones. They work with Series and the large companies that are already in the clinic. We like Bios for many reasons, and we know exactly how long will it take, how much will it cost, and we feel very comfortable with that. One, we have the data in engrafts. Two, we show the efficacy. And three, it's endogenous to the ileum. This is where it lives. This is where it works. It makes most sense. If you dose this enzyme as is, it will die on the way. So currently, there aren't any other ways, as far as I know, to deliver an enzyme, to keep it stable and to keep it alive. So we think currently we're at the best way to go, but, we think all day, so.
And with the since is the bacteria also in the ileum?
Yes.
And would it survive the acidic environment in the...
So what we see with this is about 2 weeks of washout. So we have nice engraftment for 2 weeks and efficacy. We want to see whether this is going to be a once a week or once a month dose. In terms of have a concern regarding stability, because it's already, we are basically joining the mechanism of the gut and the liver that breaks down fat. We're not dosing a molecule that is stranger to the body and you don't know what it does other than this. We're really joining the dance of bile acid circulation between the gut and the liver. So it should be stable because that's what it does, that's where it lives, in the bacteria. We are definitely looking at raising stability as well as activity.
Where's your tie to the fat here?
Where is?
The tie to the fat, so I'm sure you have data that shows that there's fat reduction?
It's not here. This is a
Okay.
colonization graph. I might have... I don't think I have that data, but I'm happy to send it to you if you're interested.
Can you show us the timeline? Like you had a slide with timeline and milestones.
This one? Yeah, so this graph basically describes the path to the clinic. And, we like planning ahead, so we broke down the time from now until we get to the clinic, how much money will we need to raise, who are we going to work with, what consultants, regulatory consultants, scientific consultants, manufacturing, toxicology. So we know what it takes and who it will take to do it with in order to get to the clinic. We're hoping to be in the clinic in less than 28 months. Of course, it depends on the mouse studies that we will run soon with our engineered enzyme. But if everything goes well, we're just following our plan.
And I'm assuming you have IP covering this enzyme?
Absolutely. We have a portfolio of IP, 2 filings that we have filed ourselves, PurposeBio fully owned, and 2 exclusive licenses from UCLA. We believe the core technology is with the PurposeBio's IP filing, and we have a lot more to file, but we're waiting until we are able to actually generate the data for the filing.
And in your phase 1, are you going to go direct to patients or do you have to do healthies first?
I'm sorry?
In your phase 1, are you going to directly go to patients, or do you have to start with healthies?
We are planning to go directly to patients. Yeah.
Great. All right, well, thank you very much.
Thank you so much.